So today what we're going to do is, finally, really, biology, okay? We're going to do the cell, and that's our selfie, right? I always say people who do selfies really have no friends. They can't ask someone to take a picture of them, they got to do it themselves, you know? So I always love that. Right? Is that true? Think about it. Like, you know, I'll take your picture. No, I'll do it myself, you know. I want to be your friend. You know? So, anyway, so we're going to do the cell, and really all this is the anatomy of the cell. I'll give you a brief synopsis of the function, but we're really going to be talking about the functions of the parts of the cell later on as we talk about how protein's made, or how DNA is replicated, and mitosis, the nucleus dividing and stuff. But this is really just and I think who here had a biology class? All right, so this is 100% of you. It's going to be more of a review kind of thing. But as you've seen the test from yesterday, just keep in mind it's not as simple as you think. All right? Sure, I might have a question, the function of the ribosome is this, A, B, C, D. But there's a lot of application stuff. There's a lot of things you apply to, or you're going to have to apply. So just think along those terms. All right, so the anatomy of the cell. So prokaryotes versus eukaryotes. All right? Prokaryotes versus eukaryotes. And the big, big, big difference is this. Can everybody see that in the back? Okay. That's the big difference. Now, they all have cell membranes. I'm not lying about that, but it's inside. Eukaryotic cells have membranes in all what we're going to call organelles, all the parts in there, whereas prokaryotic cells usually don't. They don't have a nucleus. They got a nucleoid, all right, because it's just a agglomeration of mess. It looks like a ball of hair, okay, but nothing's holding it. So everything is all happening at the same time. There's no compartments in there. Okay? That's the biggest difference. Now, there's some obvious things where prokaryotes are smaller than eukaryotes, things like that, but that's the big thing. You understand, and you know how membranes are made. If you understand what a cell membrane is that we talked about yesterday, well, the membranes around all the organelles are pretty much the same way, i.e., phospholipid bilayers. Now, certain things do have membranes around them inside. All right, most things do. Most things have a double whammy on it. There's a double membrane around them. Those are the ones that you want to be protecting really well, like the nucleus. All right, we call it a nuclear envelope, but it's really a double, I don't want to say wall, but a double membrane, because there's vital stuff in there, right? There's the DNA. So very much like, like I said before, if the United States is the cell, Washington, D.C. is the nucleus. It tells everything what to do. So there's more security in there to protect the Constitution, which is our DNA, right, for the United States. So there's certain things that have double membranes. Most have one membrane inside there. And there's going to be some that don't even have a membrane. They're just structures. We're going to learn that ribosomes, they're really not just, they're actually two structures that kind of, almost like a clam. So we've got an upper shell and a lower shell, they kind of come together. So we'll talk about that. All right? So prokaryotes, all right, they fall into the bacteria and archaea domains. Remember, the archaea domain is really one-celled organisms, bacteria, but they've adapted to a different environment. All right, they evolved more than just the regular bacteria. So, eukaryotic cells are protists, fungi, animals, and plants. Okay? Now, what's common to both of them is that both will have a plasma membrane, all right, the moat around the castle or village. Both will have this semi-fluid substance called cytosol. probably know it as cytoplasm, but here's a little difference. The cytosol, think of a pool, right? The pool has water in it. The water is the cytosol. But if you have the pool with a lot of toys in it, like beach balls and stuff, well, those beach balls and stuff are your organelles. So basically, cytosol is the water. Cytoplasm is the water with the organelles, with the exception of the nucleus. All right. So you got to. So when we say cytoplasm, it's not the same thing as cytosol, but as you get further and further with your studies, they kind of interchange that, but it's really not the same thing. And then we both will have ribosomes and chromosomes. All right, chromosomes make up genes, which we'll get into, and ribosomes will make the proteins. Okay. So a little bit more about prokaryotes. There's no nucleus, as I said. What they have is a nucleoid, which is kind of like a hairball that's just floating around there. All right, it's intermixed with everything else. There's no membrane-bound organelles in the cytoplasm bound by the plasma membrane. So it's all connected to the plasma membrane. Okay. So this is just showing you a picture of a typical prokaryotic cell. There's your little nucleoid. Now there's no membrane around that. In us, we do. You know, eukaryotic cells, we do have a membrane around there. All right. Here we have the DNA that's usually in a circle. We'll get into that a little bit later on how that actually functions. And there's other things in there. All those little dots are going to be your ribosomes. There's a cell membrane. Sometimes there's cilia. Those are those little hairs that come out to help them move from one place to another. Others might have like a little flagellum. But they're called pilia, which is basically the same thing as cilia. Now, eukaryotic cells, the DNA is found inside the nucleus. The nucleus, like I said, is a double membrane, not wall, but double membrane structure. We call it a nuclear envelope. All right? So the envelope is a double membrane area. All the organelles are membrane bound, with a few exceptions. Like I said, the ribosomes is an exception. Cytoplasm is between the cell membrane and the nucleus. Okay, so it's everything in there, including the organelles and the cytosol. Plant and animal cells have most of the same organelles. Okay, there's a couple of things that plants have, and you've probably heard of chloroplasts, and I've introduced you to the cell wall. But pretty much, plants and animals have the same thing. Do plants have mitochondria? Mitochondria are the powerhouse of the cell. Do they have mitochondria? Yeah. All right. A lot of people think, well, plants have chloroplasts and animal cells have mitochondria. No. They both have mitochondria. The plants have an added one called chloroplast, and we'll talk about that later. All right. Oh, and I don't know what I come up now, but like I said, eukaryotic cells are much larger than prokaryotic cells. So there's our typical run-of-the-mill kind of animal cell with the different parts that we'll be introducing you to. All right. You can definitely see the big nucleus there, and it has that double membrane around it. Okay? And that's a plant cell. You can see the cell wall around here. They have mitochondria, you can see here, but you have these other things that look like little stacked green pancakes. We'll talk about that later, but that's basically where chlorophyll goes inside these chloroplasts. Okay? So let's talk about, that's all I really want to say in terms of, let's say, prokaryotic cells. We'll talk a little bit about how they divide and things later on, but let's just talk about a eukaryotic cell. Okay? You got the cell membrane around it. The nucleus is over here. You're going to see what we call endoplasmic reticulum, which is basically the roads in the village. I mean, think of this as we said before, the castle in the village, right? So you have this moat that goes around this whole village, okay? inside the village, you're going to have the castle, which kind of tells everything what to do. That's going to be our nucleus. You're going to see roads all around there to get from the castle, and it's all connected somehow to the castle, right? I mean, if you look at all the roads in America, somehow they're all connected to the nucleus of America, right? Somehow, if you're in California, if you're in Alaska, with Hawaii might be a little bit different, but somehow the roads will still get you to Washington, D.C. Does that make sense? Okay? So that's our endoplasmic reticulum. Then you have little areas that are going to be building things like wood shops and food markets, things like that. Well, those are going to be our places to make proteins, which will be your ribosomes. There's going to be security in there. If there's anybody that's getting in, like a criminal, we've got to get rid of them. We have lysosomes that are going to get rid of them. So think of it like that, and it goes on. And it's probably easier to image it like that. So the cytoplasm, okay, material between the plasma membrane and the nucleus, okay? The cytosol is the actual intracellular fluid. It's mostly water. However, there's going to be a lot of solutes in there also, different salts, amino acids. They're just floating around there, okay? Think of the pool, right? You got the pool, and you have water in there, and you have big toys in there. The big toys are going to be organelles. But that water in the pool is not distilled water. There's chlorine in there, there's sodium in there, there's a lot of other things that are floating around in there that you can't see, right? Solutes. And then we have these, what we call organelles, or the toys in the pool, or the different shops and the different things that are in the village or the castle. I know I'm using two analogies here, so use whatever one works for you, okay? So we have these organelles that are the machinery of the cell, kind of making sure the village does what it needs to, whether there's schools for the children or there's security guards, there's just things that are happening over there. And each one of these organelles has a membrane. And if you understand what a phospholipid bilayer is of the cell membrane, well, each one of these organelles have their own phospholipid bilayer. Okay, it's the same thing. So if you think about it, if something can easily pass through the cell membrane to get into the cell, then that's also going to be easily getting through each one of the organelles because it's the same structure. Prokaryotic cells don't have this on their organelles. We do. Okay? Some may have two membranes, as I said. The nucleus, and we're going to talk about mitochondria. Some may not have it at all, like the ribosomes, I said. It's a big clam is what I said. Okay? So the cytoskeleton, which is basically the skeleton of the cell, what makes up its shape, the support. So it's going to help determine the shape of the cell, the support of the cell, helps organize things inside there, directs movement of substances inside the cell to make sure it goes, you have crossing guards and stuff to allow them to go on the roads and stuff inside the village. All right? So, and it also contributes to cell movement as a whole. We have certain cells that act like amoebas. We have little amoebas in us. They're called white blood cells. And they're going to phagocytize around the criminals and bring them in. How does it do that? It's the cytoskeleton that allows it to do that. Okay? So it's a series of rods running through the cytosol. I'm going to make this short but sweet. Okay, because I want to spend more time on the other stuff. But the cytoskeleton is made up of microfilaments. Microfilaments are what we call, a good example is actin. You know anything about muscle? Yes, this is where it comes in. You're going to have muscle contractility happening there, way over the scope of this course. But that's what's happening over there. It also maintains the cell shape, and this is where microvilli are there. And microvilli are going to increase the surface area. It allows for absorption. So let me just show you what this is. If this is like, if this is your wall of your intestine, all right, or I cut through your intestine, the main function of the small intestines is to absorb all the wonderful food that you're eating from McDonald's. Okay? If it doesn't absorb it, it goes down your poop scoop, right? So there's going to be a lot of absorption happening here. The small intestine is pretty long. It's about 20 feet long, which is all scrunched together. We have a lot of absorption going here. The best way to absorb things is to increase the surface area of this area. In other words, we have blood vessels that are surrounding here. And we have foods that need to go from here and be able to get through the surface of the intestines and go into the bloodstream to go where it needs to go, liver usually, and breaks up there. But I just want to concentrate here with the surface area, what that's doing, microvilli. What's happening here is that this has a certain surface area. So I'm going to just draw it bigger for you. All right? Certain surface area. What I can do, so certain things can actually get absorbed over here. But if I actually bend this up, and these are called villi, we're still not big yet, or we're still not small. Now, yes, I can still absorb here and absorb here, but look, I've created new surface area here. See that? I've increased the surface area. Now, microvilli is so small you can't even see with a light microscope. Microvilli is that we're going to add, look, I'm just drawing it bigger on here so you can see it, but you're going to add these microvilli all around here. So look what's happening. Let me just draw it again over here so you can see it. What's happening is, before you could absorb things here, you could absorb things here. But now look, you can absorb things here, here, here. Look at this. Look at what the surface area is doing. You've created much bigger surface area so that we can have major absorption going on. See that? That's what, keep in mind with surface areas. It's a common thing and a common word that you're going to be hearing for the rest of your career in science. But make sure you got it down. It has nothing to do with surface tension. Okay? But they'll be using it and they're going to assume you know what the surface area is. So that's why I'm trying to emphasize what it is over here. So microvilli, and I'm emphasizing also because microvilli is increasing surface area. And they look like these little hairs. But I don't want you to get mixed up with something that looks similar that does something totally different. Cilia. Okay? And we'll get into that too. We also have intermediate filaments that's also going to strengthen this nuclear envelope and also maintain the cell shape. And we also have microtubules that organize organelles that move cilia and flagellum and will also aid in cell division. Cilia is totally different than microvilli. It's a very common thing that students have in terms of problems. So if you have, let's say, a bacteria over here, there's these little hair-like things that kind of look like microvilli. The difference is microvilli just stay like that. So absorption goes on. Cilia move. And it helps, the way that they work, it helps move this critter forward or backwards, whatever. So do make sure, hint, you know the difference between cilia and microvilli. All right? Look alike, but they're totally different. Okay? So this is parts of the cytoskeleton, a lot of different functions of them, okay? And this is trying to show you the different, microvilli, or the cytoskeleton, how it looks inside the cell. All right, so let's talk about centrioles and spindle fibers. Okay? These are located in what we call the central zone. And a central zone is an area that's made up of two, what we call centrioles. MUO is made up of. And they kind of go on a 90-degree angle on each side. So one centriole will go this way, the other one will go this way. When you put the two together, it's called a central zone. Okay? It's at the center of the microtubular formation, and basically they're also found at the bases of the cilia and flagellum. To get that, that's like where the motor is for the flagellum and the cilia. That's what actually makes the thing move. We'll also be talking about it in terms of mitosis and how that actually will cause what we call these mitotic spindles, so that we could actually make the two cells divide. Okay? So that's what they kind of look like. That's this centriole here and a centriole over here. You put it together, and that's the whole thing is a centrosome. And there's two of these, two sets. So there's one here and one on the other side. And you're going to see, let's say, when we do mitosis, they're going to move to opposite poles of the nucleus. We'll get to that later. So cilia, all right? Let's talk a little bit about that. Cilia. Now, most cilia will move, but there is some cilia that it doesn't move on its own. It will move, but something's got to make it move. We have cilia actually in the nerve parts of our ears. And they just sit there. We've got a lot of fluid in our ears. So what happens is when we turn our head, the fluid in our ears are going to bend this. Kind of like if you've seen an aquarium and you have the sea anemone in there, you know what I mean? I don't know the genus, the species name, you know. But it just sits there. But if you tap the aquarium tank, they kind of move, right? But they usually just sit there. Does that make sense? You've seen those tentacle-like things, right? The sea anemone kind of thing. So that's what I'm saying, is they don't move by themselves, but something will move them, and then that'll do a chain reaction and send a nerve impulse to your brain and say, hey, I just heard something. Then we also have cilia that you do move, all right? They beat in waves, and we see this in our respiratory tract, all right? In our trachea, our windpipe. And they beat. So when you have a lot of gunk going there, that's a good medical word, right? Gunk. All right. But when you have a lot of gunk, the bacteria that's floating in there, there's mucus that's just building up. But what's going to happen is the cilia is going to push it in a certain way where it's going to push it towards your mouth, and it also develops a cough, so you can cough everything up. We also have cilia in the fallopian tubes of females, where you have the fallopian tube. Our Dodge Ram. Right? Sorry, that's female anatomy. This here is the vagina. Here's the womb that holds the baby. The two fallopian tubes go this way. Here's the ovaries with the eggs. Let me orient you, and then I'll show you what's going on over here. An egg pops out of here and moves into the fallopian tube. All right? Penis goes here. Sperm will go this way, through into the uterus or womb. This is connected. Sperm goes this way. They unite over here. So the egg is coming out of the ovary and goes this way. They unite this way, and then it takes about 7 days, and then it moves back from here. So one thing about this, not really talking about this, is that the penis never goes where the baby is. Does that make sense? The baby's going to grow over here. You've got the little embryo growing in here. Okay? But what I want to say here is that in the fallopian tube, we have cilia, and they move towards the womb. Does that make sense? All right? So it's pushing the egg towards the womb. And the sperm goes over there. So it's like the cilia saying, go ahead, he's a nice guy. Get ahead, keep him going. He's a nice guy. You go meet up with him. It's like the cilia is like a little Cupid. Right? So that's what's going on over here. So cilia is in different places. And also a lot of one-celled organisms have cilia to help them move, okay? So we don't see it just on one-celled organisms. It's all over your body also, okay? But they're beating, they're moving that way. So it's kind of like it goes down, comes back up like this, and whips it again. And that's how cilia usually works. Okay? So look what's going to happen here. These cells over here have cilia up on top. Now, if there is a lot of mucus here, so we're talking about, let's say, in the trachea, in our windpipe. If there's a lot of mucus here, the cilia is going to push that mucus upstream to where your mouth is, produce a cough, and you cough out all that mucus. That's how it goes on over here. At the apex layer, in other words, let me show you here. This is the apex layer. This is the basal layer. This is where it's attached to some underlying tissue. But this is the free end. This is where we call it the lumen. This is the area. When you cut open, let's say, a garden hose, the water that it goes through that canal is, that cavity is called a lumen. Any open cavity in a tubular structure is called a lumen, L-U-M-E-N. Okay, so you'll hear that even in a few things we're going to talk about later on. So the apex of the end, or the end of the, this is the apical end of the apex, that's the part that's going over here. Okay, it's the part that's actually in contact with that lumen. So what's happening here is this, how cilia works. And there's a point to this. Saline layer. We have chloride pumps that are pushing chloride out there. Now, just put your pencils down. Think about what's happening here. The chloride is being pumped out there. Is chloride negative or positive? Negative. Okay? So it's going out there. But it's making the lumen area more negative or more positive? More positive. Okay? Is it more positive? No. If it's putting more chloride out there, it's more negative. But that's kind of unstable. We don't want that. So we're going, because negative is going out there, we've got to put a positive out there too so it stays neutral. Does that make sense? Right? So what happens is, as chloride is being pumped out there, sodium, which is a positive charge, is also going to get pumped out there. Well, it's not really pumped, it's just because we're trying to make it stable. Now it's perfect. The charge is perfect, right? Zero. You got negative out there, you got positive. But we got a problem, Houston. When you put more salt out there, isn't it going to be more salty or more concentrated? Not good. That's not stable either. So get a load of this. After the chloride gets pumped out there, sodium's going to follow, but water is also going to follow. Because for every sodium and chloride that's going out there, it's making it salty out there, we got to also send a molecule of water out there also, so that we can keep it isotonic to a point. Does that make sense? So you're diluting it. But look what's happening. So are you putting more water out there? Yeah. So it makes for a saline area, an aqueous area. It's breaking down the mucus because you're putting more water out there, all because of a chloride pump. Does this make sense? Can you visualize this? Let me reiterate, because I want to show you what's going to happen afterwards. Chloride gets pumped out there, active transport. When we see the word pump, it means we're active transport, we're going against the gradient. We're putting chloride out there. That makes it more negative on the outside. So sodium is going to go over there so that the charge is good. But now we got more salts out there. It's going to be a hypertonic solution. So now water's going to go out there and dilute it. And that extra water that's going out there is going to break up the mucus to become more of a liquid, more of a saline or aqueous solution. It's not going to be mucus anymore, it's going to be watery. You don't want all that mucus in there. You're going to have problems breathing. You need it to be water so you could actually, that's easier to cough out than mucus. Does that make sense? It's a whole cascade of events or domino effect going on. But this is also a running theme that you're going to see later on with a lot of things. When something goes out there, the sodium's going out there, water's going to follow. Okay? So that brings up cystic fibrosis. Who here has heard of cystic fibrosis? All right, more than half. But you might not know what it is. You might know how the person is behaving with cystic fibrosis. They're coughing up and they're having problems breathing. Let me show you what's going on. There's a defect to that chloride pump. That chloride pump is not pumping. They're there, they're just not pumping out chloride. So if you're not going to pump out chloride, right, is sodium going to go out there? No. Is water going to go out there? No. So what's the fluid going to be like inside, let's say, the trachea or windpipe? Is it going to be mucousy or is it going to be liquidy? Mucousy, right. And that mucus sits in there. Now here's a little thing. The mucus is a wonderful medium for bacteria to grow. They love it. They go party hardy in mucus. So these people with cystic fibrosis have many, many infections of the lungs. And that's what they usually die of. All right? So it's a hereditary disease. 1 in 20 Caucasians carry this gene. We kind of routinely, now we're kind of routinely just seeing if you have this. All right? The chloride pumps don't work, which means you're not going to get this adequate saline fluid out there. All right? You get these thick mucus plugs in both the respiratory and a lot of places. Okay? So it's inadequate digestion of nutrition, absorption of oxygen, and they usually will have a lot of infections. Okay? That's usually with the diet. 1 in 20 Caucasians carry this. Okay? The bad thing is life expectancy is only 30 years old. So it's a pretty serious thing. Okay? There's a lot of lung treatments, respiratory treatments, where they teach the parents how to beat on the back to try and break up the mucus, and then teach the child how to cough it all out. It's kind of difficult. They have a lot of respiratory infections. Questions on that? All right? I try to throw in, some kind of medical or some kind of contemporary issue whenever it's possible. Bring back some feedback. Okay, flagellum. All right? It's similar to the cilia, except longer. And there's only one cell in the human body that has that, and the guys have it, and the girls don't. It's a sperm. Okay? So it works basically the same way as the cilia, except it's